Okay, good evening, ladies and gentlemen.
Good evening.
Yeah, well, we are back again. Um, how has how are the lessons going?
We are going okay
Okay.
with new courses being introduced.
Oh, why is that?
Well, I think they've introduced two new courses that will have to even sit for the exams. So, yeah, we are trying to catch up.
Okay. Um. Okay. So let me just stop presenting and see. Okay. So can you see my slides? Can you see the slides and can you see them move?
No, no.
You can't see them. Not even now.
I
We
can.
can now. Now we can.
Oh, now you can. Now, okay. So, ladies and gentlemen, we are looking at muscle physiology. I'm sure that you have gone through this. The only trouble is that that recorded lecture, at the end, it sort of cuts. And I'm recording this one in case you need it for your future reference. So, basically, we are going to talk about, um, the skeletal muscle anatomy. We are going to talk about excitation, contraction coupling. We're going to talk about the sliding filament theory. We're going to talk about muscle mechanics and a little bit on muscle metabolism. So basically, as you know already, there are three types of muscles in the body. So there's skeletal smooth and there's cardiac. To put it loosely, the skeletal muscles are obviously the voluntary muscles of the body. They are responsible for limb movement. They help us with chewing, swallowing, breathing, etc. And they're also important in generating heat. Um, you know, when shivering, we are going to talk about that when we talk about the integumentary system. And also of note is the fact that these muscles are voluntary. So for them to move, you have to consciously, instruct them to make a movement. We also have smooth muscles. These are involuntary and they'll be found in most internal organs of the body, namely. So you find them in the blood vessels, you find them in the GIT, you find them in the urinary tract system, etc. The fact that they're involuntary, so they're basically, you know, you don't really have to consciously think about them for them, to contract. We also have the cardiac muscles. Basically, they're the muscles that line, the heart. They're also involuntary and they're responsible for the pumping action of the heart. I need to tell you, in the previous exam, there was a question on classification of muscles. So it was simply based on this. What you're looking at, ladies and gentlemen, is basically a histological slide of the skeletal muscles, cardiac muscles, and the smooth muscles. So you know what histology is. So you know you have these pathologists, and what they basically do is that they take small samples from the body and they prepare these samples in such a way that they can look at them, under a microscope. So basically, look at body tissues at microscopic level. If you can see my arrow, what we are looking at here is basically what you see in the skeletal muscle. You will note how cylindrical. So these are basically the skeletal muscle cells themselves. So it is a muscle and it is striped, or better described as striated. Okay. So you will note also the fact that these little pink structures on the periphery, this is actually the nucleus of this cell. And the interesting thing is about muscle cells is that they may have one nucleus for a particular for one cell. You may find more than just one. We are used to finding one nucleus per cell, but these particular cells may have more than one at a particular time. Um, I've already mentioned the fact that they look like zebras, only of course it is not black and white. And you will not, as we discuss, this striped nature is as a result of the arrangement of what is known as the sarcomere, as you are going to see later in our discussion. Next up, we have the cardiac muscles. If you look closely, they do have very loose areas that look more pink than others, but it is not as well arranged, these pink areas that we are looking at, as the skeletal muscles. So, because they do have these pink areas in them, they are also considered as striated as well. More interestingly about these cardiac muscles is the fact that they are interconnected. So basically, they have what is known as intercalated discs. These intercalated discs are important in that they connect these muscles to the next muscle mechanically through what are known as desmosomes in the intercalated discs. But also there's an electrical connection so that the ions can move from one cell to the other in what are known as are they called gap junctions? Sorry, I forgot the name. I'll tell you when I remember. But basically, there are special channels that allow ions to move from one cell to the other, and it helps to spread the depolarization, basically. Next up,
Excuse me,
yes,
sir.
please.
The Caesar is trying to join the meeting. The Caesar is trying to join the meeting.
Okay, one moment. All right. So Cesio has been admitted. So next up, we have what are known as smooth muscles. Interesting thing about the smooth muscles is that they do not have this striped nature that you see both in the cardiac muscles and in the skeletal muscles. They usually have one nucleus, per cell, and it is central as opposed to the periphery nature that we see in the skeletal muscle, basically. In this next slide, what we are looking at is just basically a skeletal muscle and a low power light microscope. So under a microscope, so you can see they appear cylindrical. So each of these cylindrical appearing structures are basically the cells of the skeletal muscles, and you can see that you've got this pink peripheral structures, which is basically the nucleus of these particular cells. Okay, as we've mentioned already, so we're going to focus a little bit on the skeletal muscles, then the cardiac and the smooth muscle. So functions of the skeletal muscle, as you know already, they're important in movement, they're important in helping us maintain posture and body movement, they're important in helping us to stabilize joints, and of course, they're important in generating heat as and when required. So I'm talking about in an instance where it gets cold, and I'm sure you've seen from the notes that I provided as a way of maintaining a constant body temperature. If, let's say, the external environment gets cold, the body is going to put up measures to try and generate heat, and one of them is the involuntary contraction of muscles, also known as shivering. In particular, the skeletal muscles themselves. This is the only time the contraction is involuntary, and this contraction of the muscles is going to help the body to generate the much needed heat. So, we can talk a little bit about the organization of a muscle. We know that a muscle in itself is made up of several muscle cells, and these several muscle cells, when grouped together, they are known as muscle fibers. Um, these in a particular muscle cell, so if we were to go into a muscle cell, we'll find a special adapted fibers, which are called myofibrils. And these myofibrils are made up of special proteins, namely myosin and actin. And these myosin and actin, are arranged in a special fashion such that they overlap each other. And this special fashion is called a sarcomere. And the sarcomere is actually, the unit of contraction for a muscle. As I'm going to show you later in this discussion. So in this slide, I'm basically showing you. So we've got in the picture a muscle tissue, and at the end of this muscle tissue, we've got tendons. We know that most of these muscles are going to attach either to another muscle or bone via a tendon. And in this image at the bottom, so you see that um, basically you have a whole muscle tissue which has been magnified, and the muscle tissue will then will be formed by a group of muscle cells. You can see that these muscle cells, they form a bundle, and this bundle then will be grouped together to form the muscle tissue itself. So here I'm basically talking about same thing. So we've got the muscle fiber, which is the cell itself. So the interesting thing is that the cell is going to have some, connective tissue that is going to cover it and separate it from the other muscle fibers or muscle cells in a bundle. But even in a bundle, there will be a special sheath that is going to separate one bundle from the other bundles. And eventually, a muscle cell, as in a muscle tissue, is also going to have special connective tissue. And I've got the names of this connective tissue listed in this diagram. So what you see at the bottom here is basically a muscle fiber or a muscle cell that we described. So you can see that this whole thing is the muscle cell. You can see that at the periphery, you've got your nucleus, and you can see this striped nature that you're looking at. If you're to magnify it further, you're going to see that these striped things that you are seeing is basically because of the arrangement of the actin and myosin that we talked about in what is known as a sarcomere. I'll give a little bit more detail as we. Right. So this actin and myosin, like we've already said, are the contractile proteins that you're going to find in the myofibril. We did say that the myofibril is basically the components in a muscle cell that are responsible for, helping a muscle contract. And we did say that at molecular level, this myofibril is made up of actin and myosin. So this actin and myosin, basically, we did say that they are proteins. Of the two, the myosin is the thicker one. The actin is the thinner one. So, does somebody have a question?
Not a question. Sizwo is still trying to log in, but you haven't yet let him in.
That is strange because I did accept. Let's see if he went out. Well, I don't see anybody trying to join. No, there's no one. I don't know what's happening to Caesar. Okay. Okay. Welcome, Mr. Sizwe.
Thank you so much. Though.
Okay, guys. So as I'm talking, you can see my slides move, yeah?
Yes, we
Yes.
can.
Okay, that's very good. So we are talking about this sarcomere. So this sarcomere, like we already said, is made up of these proteins, which is actin and the myosin. Now, we did say that it is the actin and the myosin that makes up a myofibril, which we did say is like a filament which helps a muscle cell to shorten and therefore contract. So how does that happen? That's what we are going to look at. So this, and we did say that the myosin is a thick protein, a filament, while the actin is the thinner one. So the spatial arrangement that we're talking about, you can see that there's some sort of overlap between this actin and myosin. So what basically happens when a muscle contracts is that the myosin will not move, but it is going to pull this actin towards the center, which is called the M line. It's got components. I'm not going to go into the details of it. I've given an explanation in this writing. You can go through it on its own, but I'll make reference, to some of them. So basically, the center of this myosin is got the M line. So what happens is that when a muscle contracts, it's going to pull this actin towards the center, which is the M line. So you can see that if this myosin is pulling this actin this way, and this actin is being pulled that way. So basically, you're going to have shortening of the sarcomere, and you are going to end up with shortening of the myofibril. Eventually, you're going to end up with shortening of the muscle cell itself. And if several muscle cells do shorten, you're going to have shortening of the muscle itself, which will result in some sort of movement. We shall talk details as we go. So we've already said of the two proteins, the myosin are the thicker ones and the actin are the thinner ones. So the interesting thing about this myosin is that there is they have got heads. So these are the thick filaments of myosin that we are talking about. And you can see that they've got these heads. So on the surface of these heads, you've got actin binding sites, and you've got you could say a binding site for ATP as well. And I'm going to explain the importance of this. So we did say that the actin are the thin ones. So just like the myosin has got binding site for actin, actin also has binding sites for the myosin. And allow me to explain as we go. So apart from just this actin and myosin, you've got two additional proteins which act as regulators, or if you like, regulatory proteins. So these proteins are namely tropomyosin and troponin. You can see in this structure, I have these little bluish looking structures, which is what we said was actin. So what you're going to have in the sarcomere is that this actin is going to have a certain protein that is going to cover the binding sites of myosin, and this protein is known as tropomyosin, as you can see. Additionally, this tropomyosin is going to be held into place by another special protein, which is called troponin. So the troponin helps to hold the tropomyosin over the binding sites of the myosin binding sites on actin. It sort of covers them, and you're going to see how all this thing works out. So in this structure is just basically a pictorial summary of what we have discussed. We have discussed that we've got myosin, we've got actin. We said that the myosin has got heads, and that these heads that bind to on the actin on the myosin binding sites. And we said that there are two important regulator proteins, namely the tropomyosin, which covers the binding sites, and the tropomyosin is held into place by another special protein known as troponin. Absolutely. So moving on. So this basically another again is a pictorial representation of the sarcomere that we've talked about, which I believe now you are experts. What you're looking at is an actual electron microscope view of the sarcomere, and you can see that in the center we've got overlapping of the actin and myosin, and at the end here we've got what is known as the Z line. So basically that is the Z line. And I'm going to show you in the structure what the Z line is. So if you look at this simplified myosin, so you can see that the Z line is basically the end of the sarcomere. So if this is the left end of the sarcomere on the Z line, that would be the right end of your sarcomere, basically. Okay, moving on. So we've already said that since the sarcomere is the contractile unit of a muscle, we said that what will happen, the summary of events, is that you need the myosin to pull the actin towards the center of the sarcomere. That will lead to shortening of the sarcomere, that will lead to shortening of your myofibril, that will eventually lead to shortening of the muscle fiber, which is the muscle cell. And if several muscle fibers or muscle cells shorten, you are going to eventually end up with shortening of a muscle, and that is how you are going to produce movement. So basically, all these slides are talking about that. I'm going to skip them. So now we want to understand how that shortening comes about. Is it magic or is it witchcraft? We want to understand the science behind that. What are the processes that lead to the shortening of a sarcomere that eventually lead to shortening of a muscle, which helps us to produce movement? As you may know, ladies and gentlemen, unlike cardiac muscles, which are able to generate their own action potential, muscle cells do not have this capability. So a muscle cell cannot self-excite. It relies on the nervous system to do that for them. So as you may know, our muscle cells have a nervation. So that is, they are connected to the nervous system. And they are, the, the connection between the nervous system and the muscle fibers is called gap. Oh, that is a mistake. It's a neuromuscular junction. Oh, by the way, now I remember because I thought I was confusing the two. So I told you that in a cardiac cell, you have the cells being connected to each other, as opposed to the skeletal cells, which are basically linear or parallel to each other. The cells in the cardiac cell, or rather in the cardiac tissue, will sort of connect with each other in what are known as intercalated discs. And the intercalated discs has two junctions. You've got desmosomes, which are basically the mechanical connection between the two, and you've got gap junctions. It is the gap junctions which allows the ions to move from one cell to the other. This is important so that if one cell is excited in the heart, that wave of depolarization can easily spread to the next cell. Why is that important? So that the muscle then can contract as one unit, as a syncytium. We are going to have this discussion next semester, of course, if I'll still be an employee of this university. All right, so we're trying to understand basically the summary of events that bring about the shortening of the sarcomere. To put it very loosely, the summary of events is that you are going to have the action potential, which is going to come from a nerve, okay, that connects to this muscle fiber. When this action potential reaches the neuromuscular junction, which we said is the connection between the nervous system and the muscle fiber, there is going to be release of acetylcholine. I'm sure you're familiar with that. The release of acetylcholine, when it is exposed to the membrane of the muscle, there's going to be a series of events, but basically it's going to, lead to, the release of calcium. I'm going to give you details later. This release of calcium is going to activate the troponin. Now remember, the troponin is a regulator protein. Just trying to refresh your memory. You see these soybeans, yellowish looking structures that are holding this tropomyosin in place. We said the tropomyosin is like a belt that is covering the binding sites of myosin on actin. So this troponin hold it in place so that it doesn't move. So what will happen after the depolarization from the action potential from the nerve? It will lead to the release of calcium. The release of calcium is then going to cause these little structures to be activated, and when they're activated, they're going to release the tropomyosin. So basically, this tropomyosin is going to come off the binding site on actin. When it comes off the binding site, what happens next? When it comes off the binding site, what will happen is that now the binding sites for myosin are free. So those heads for the myosin can now go and bind there. When the binding has happened, we say that cross bridges have been formed between the actin and the myosin. You are going to see details a little bit later. So once the cross bridges are formed between the actin and the myosin using ATP, which is a quanta or quantum of energy for the body. This myosin can now pull the actin inwards or towards the M line on both sides, which then leads to the shortening of your sarcomere. And that action of pulling the actin on both sides towards the M line is called the power stroke. Once the shortening has happened, once the power stroke has happened, there's no need for the myosin to bind there anymore. So what will happen is that now the myosin and the actin need to come off. Unfortunately, it is not possible to come off. It is a process that requires, again, ATP in what we say is the reset process. Okay? So this is basically the summary of events that happen in the excitation and contraction coupling in a sarcomere. But I'm going to give you much more details just on that. So just to give you more details on the steps again that I did summarize earlier. So we did say, ladies and gentlemen, a muscle cannot contract on itself. It depends on the central nervous system to give it an action potential at the neuromuscular junction. So as you can see, what we have here is basically the spinal cord, and we've got these motor neurons that are innervating the nerves, or rather the muscle fibers. So an action potential is going to come from the neurons that is going to be spread to the muscle fibers that will result in shortening of these muscle fibers as a result of the contraction of the sarcomere. Right, are we still together?
Yeah.
Yes, we are together.
Now you guys are in service, so these structures are not new to you. So this image is also just a drawing of what the neuromuscular junction is. So as you can see, this is the terminal end of a nerve. And at the terminal end of a nerve, you will have vesicles that are going to have a neurotransmitter. In this particular case, it's acetylcholine. And once an action potential reaches this terminal end, you're going to have exocytosis of this acetylcholine in the synaptic cleft. And it is this acetylcholine which is then going to activate the entry of sodium into your muscle fiber and exit of potassium from your muscle fiber, which will then result in the release of calcium. And we said that the calcium is going to activate the troponin. The troponin is then going to let loose of the tropomyosin, and then the binding sites for the myosin is going to be open. The myosin is going to bind, the power stroke is going to happen, and then there's going to be a reset. Okay, so we've talked about the first step, which is basically the arrival of the action potential from a nerve. The second step is the release of calcium. I've already told you already. So in a muscle cell or muscle fiber, the endoplasmic reticulum, which is an important organelle in a cell, as you know, important, in the manufacture of proteins. In a muscle cell, they are specially adapted to store calcium, and they are no longer called endoplasmic reticulum. They will be called sarcoplasmic reticulum. So, with the arrival of the action potential, the sarcoplasmic reticulum are going to release calcium. Okay. Like we've already said, once the calcium is released, we go into the third step. It will lead to activation of the troponin. We did say that the troponin is a regulator protein, which is what holds The troponin are the yellowish looking structures, and this strap-like looking structure is a tropomyosin, and this is it green or bluish looking structures are basically the actin. And you can see that this belt looking structure is covering what look like what you would see as the eye of the soybeans, like I like to call it. Yes, it's because I'm a soybeans farmer, or a farmer wannabe, if you like. And you can see that the yellowish structures, after being activated by the calcium, the tropomyosin, which is a belt-looking structure, is coming off, the eyes of the soybeans, or the binding sites of myosin. And if the binding sites of myosin are free, you can see this reddish or pink-looking structure coming to bind there. This is actually the myosin itself. Okay, that takes us into the fourth step. Once the myosin binds to its site on the actin, um, it forms what we call cross bridges. Okay, and once the cross bridges are formed, sorry guys, I don't know what I have missed. Okay, so I think so in the fourth step, this is where we have the tropomyosin moving out of the way, and we've got the myosin actually binding on the site of actin. When it binds, it forms the cross bridges in the fifth step. And in the sixth step, what will happen is that so this is the just after forming the cross bridges, there is no movement here, or rather, this is the beginning point. But you can see that with the help of ATP, this little head of the myosin contracts, pushing the actin in that direction. And this is basically what causes the shortening of the sarcomere, which is eventually what will cause the shortening of a muscle cell or the muscle tissue in itself. Now, after the binding, after the and this contraction by the myosin is known as power stroke. Now, after the power stroke, this myosin and actin will still be bound together. To But if they're still bound together, then it won't be possible for the muscle to generate another shortening. So the actin and the myosin needs to unbind. And for it to unbind, it needs to use ATP again. Okay? In fact, maybe I should pause to ask you, do you know the scientific explanation why when people die approximately about 2 hours after death, the muscles start to become stiff, or what is known as rigor mortis? Mr. Sizwe, sir.
Yes, Doc.
Mm,
I have no idea on that one.
Mr.
I might lie. I have no
Which
idea.
is your hospital, Mr. Sizwe? We need to tell the SMS.
No, I only know it. I haven't taken time to find out, especially on the physiology, on what really happened during diagnosis.
For your information, we had an MCQ like this. Can any other one tell us, Madam Temba?
What's the question, sir?
Yeah. You're not even with us. That is the problem of online classes. Can you tell us?
I missed the question. No, I'm following, but I missed the
Okay,
question.
we shall tell you. Can you tell us the explanation behind rigor mortis with what we have learned?
I think okay. From what we have learned, there's no because there's stiffness of the muscles. There's no movement because everything has stopped working. So there's no activation on the neural what? I've forgotten.
Neuromuscular junction.
Neural muscle something. Yes, there's no activation on there's no message being taken to the neuromuscular junction. So hence calcium is not being released for activation of the muscles. I think that's what I can say. Hence the stiffness.
Any other? Okay, I'll tell you. So, guys, Madam Timber, so you are incorrect because, well, but you are partly correct, but your answer doesn't really hit the nail on the head. So, of course, when somebody is dead, there's no more action potentials that are reaching the muscle fibers. But the truth of the matter is that there will be a lot of calcium that is going to be released. Why is there going to be a lot of calcium? You see, when you are alive and the cells are alive, they work so hard to keep a lot of potassium inside the cell and a lot of sodium outside the cell. But when you die, that concentration gradient is lost, and that goes for calcium as well. So you're going to have a lot of ions everywhere. So because of this, there will be a lot of, let's say, calcium everywhere. We can assume that there will be activation of the special proteins that we spoke about. However, there will be no power stroke. You understand? Because there is no ATP. You're not breathing oxygen. You need oxygen and the metabolic body processes to be working for you to generate ATP, which is the currency of energy for the body. And remember, you also need ATP for the myosin and the actin to be free. But because it's not there anymore, so the myosin and the actin are going to hold on to each other and the muscle will become stiff. That's basically how your rigor mortis comes about. You get, madam?
Yes, sir.
So the calcium is not, or rather the ATP is not only important for the actin and myosin to reset, that is to come loose from each other, but it is also important for the calcium which was released by the activation, of action potential to go back into storage. So to go back into the sarcoplasmic reticulum. You understand? So the ATP is going to do two things. So, ladies and gentlemen, a summary of what we have talked about. These bluish looking structures that you are looking at is basically the actin, and at the bottom we've got the myosin, and these little things that you are looking at is basically the heads of the myosin. And we said that after all the chemistry, action potential arrives, release of calcium, activation of troponin, um, removing of the tropomyosin from the heads, from the myosin binding sites on actin. We said that these heads are going to bind, and when they bind, they're going to form cross bridges. After they form cross bridges, using ATP, they're going to power stroke, and you can see when they power stroke, they're going to pull in the direction of the center, and they are going to lead to the shortening of the sarcomere, which is eventually what is going to lead to the shortening of the myofibril, then the shortening of your muscle fiber or muscle cell, and eventually the shortening of the muscle itself. Okay, does it make sense?
Yes, it does.
Yes, it does.
Absolutely, guys. Okay, guys, I'd like to briefly tell you about the sliding filament theory. And the sliding filament theory is basically from the step 3 to step 6. So this basically the events that are going to happen, from the activation of the troponin to the time that your myosin is going to pull the actin towards the M line that will lead to the shortening of the sarcomere. So it's a very simple concept. Gonna skip this. We can talk about phases of muscle contraction. So we've got latent period, and then we've got the period of contraction itself, and we've got the period of relaxation. So the latent period basically is the time it takes, for the for your action potential to arrive at the neuromuscular junction, and the time it will take for all that calcium to be released, for all that activation of the troponin and all that kind of stuff. And it is in milliseconds. Okay. Yeah, basically. The rest you can read. Okay, so what can we talk about? Okay, we can skip this one as well. Okay, so a little bit on muscle mechanics. So we did say that in a particular muscle, there are going to be several muscle fibers, which when they contract, they're going to create movement. We did already talk about that these muscles are going to be surrounded by connective tissue. It is this connective tissue which will extend at the terminal ends of these muscle fibers to form tendons. And it is these tendons then that are going to be attached either to other muscles or soft tissue or bone. So when your muscle contracts, it's going to create what is known as tension. And when this tension is created, it will cause basically to stretch the tendon. And if the tendon, wherever this tendon is applied, either to bone, another muscle, or any other soft tissue, it is going to apply a force wherever it is attached, which will then cause movement. Let us say an example: if we have the biceps, which is attached to the, radius or ulna, contraction of the biceps is going to help us move these two bones upwards against gravity. Or if I was carrying something in my hand, which would be referred as a load, then it will help me to move that load against gravity. Okay, it's basically what we've talked about. So I can briefly tell you about the types of contraction. So you basically have what is known as concentric contraction. In concentric contraction is that the muscle contraction will, when the muscle contracts, it becomes shorter and tension increases. An example of concentric contraction is the lifting of a weight as we have described. So the biceps will become shorter to help you lift that weight. And as it, becomes shorter, its tension will increase. You also have what is known as eccentric contraction. So here, basically, what will happen is that, the muscle will contract, the tension increases. However, the length of your muscle increases. So an example of that is when you let's say if you're a weightlifter, you've been lifting the weight, but now you're putting it down. So your arms are stretching, so your biceps, instead of contracting, there's tension, but it is lengthening as you are putting your dumbbells down. You also have what is known as isotonic contraction. By the way, the prefix iso means same. So if they say isotonic, it means same tone. So meaning that the tone is not changing. So an example of or rather, what happens in isotonic contraction is that when the load remains constant, the muscle shortens, but the tension remains the same. So the tension remains the same, but the muscle shortens. An example is when you're shrugging your shoulders. So we know that, when you shrug your shoulders, the muscle that is responsible for that is your trapezius. So you also have what is known as isometric contraction. We did say iso is same, metric is length. So isometric, so same length. So the length of the fiber is not changing. So what happens here? The tension would develop, but bone and load is not moved. So an example of this would be if, let's say, you're trying to lift, a very heavy object, which is beyond your capability. There's tension in your muscle, but there is no movement. The bone will not move and the load will not move. Absolutely. Okay, so would like to talk about some of the factors that influence muscle tension. So what are these? So you've got the motor unit recruitment, you've got the size of the muscle and the number. So the size and the number of muscle fibers, we should say it like that. Muscle twitch summation, the Trepe effect, and the optimal length of the muscle fiber. I'm going to give details. So a motor unit, ladies and gentlemen, is basically the nerve, you know. So it is the nerve and the muscles that it supplies. So the nerve and the muscle fibers it supplies are known as a motor unit. A motor unit is very different from a neuromuscular junction. I think I've seen some people confuse this too. So so basically, why is this important? Why are we talking about it? We are saying that because we are looking at factors that influence muscle tension. So we know that in a muscle, the more motor units we recruit to do a particular task, the more tension we are going to have in that particular muscle. An example would be, if you're using your biceps and you are lifting a light load, let's say you are lifting maybe a cup, the motor units you're going to recruit to do that task are not going to be the same as you would recruit if you are lifting a bucket of water. You understand? So to lift a bucket of water, you need more tension. So you will need to recruit more motor units to carry out that task. Okay, so we've basically described the concept of the motor unit. So I'm going to skip this one. I think we've already gave examples already, but we also know that the size of the muscle and what determines size of the muscle, it is the size and number of its fibers. So the more the fibers in a muscle and the bigger they are, the more tension you can build basically in that particular muscle. And we know that the more one exercises, the more one builds muscle bulk, and that comes about because the muscle fibers increase in number and they increase in size. Basically, we also know that the opposite is true. If you don't exercise, you will have muscle atrophy. Why do you have muscle atrophy? It's simply because the number and size of your muscle fibers will reduce. Okay, we can now talk about muscle twitch summation. I find the best way to understand this muscle twitch summation is basically to look at this graph on the right. So in blue, what I have is the response of the muscle. So in the y-axis of this blue plot is tension, relative tension, and on the x-axis, what I have is time. The the buildup of tension, or rather the change in tension in our experimental muscle that we are plotting, is related to this red plot, which is basically the action potential. And you can see that on the y-axis, there's voltage here. So what we are saying is that when an action potential arrives at a muscle is going to respond by contracting, by doing all those things that we went through, the tropomyosin, the actin, and all those things, eh? So you are going to it result in muscle contracting, but as it contracts, it's going to build up tension. And after it contracts, it's going to relax and the tension goes down to zero from where it started. And after it rests, if you bring another action potential, the muscle is going to respond by contracting again. And when it contracts, it builds up tension and then relaxes, and so on and so forth. But what if you bring an action potential, to this our muscle, so you bring an action potential, the first one, the muscle contracts, the tension increases, and it starts to relax. But just before it finishes relaxation, you bring another action potential. What will happen? So, what will happen is that the muscle is going to contract even further, increasing tension in this muscle even further, as you can see on the plot before it finally relaxes. This effect of building up tension even more than what was as a result of the first contraction is what is known as twitch summation, ladies and gentlemen. The response of the muscle by building, attention as a result of a single stimulation is basically called a single twitch. But this one, where you have additional buildup of tension on the first one, because you have an action potential arriving at the muscle before relaxing, this one is called twitch summation. You get it, guys? Are we together?
Yes.
So you don't get it. Is it difficult?
No,
It's not them.
no, at least I'm able to understand.
Class rep, what do you think?
I had dropped off at some certain point. I couldn't get you, so I just rejoined. But I think it's okay.
Okay, the good news is that I'm recording this. I can give you a recorded version when I meet you, hopefully tomorrow. Then you can revise it before we have a test.
Oh, really appreciate that.
Absolutely. Moving on. So, apart from twitch summation, ladies and gentlemen, remember what we said? In twitch summation, what is happening is that the muscle is starting to contract and then it gets an action potential coming, as you can see in this plot. And then you've got an additional buildup of tension in the muscle. But what if, you have action potential that are arriving at the muscle without giving it any chance to relax, what are you going to have? So you are going to have sustained maximal contraction of the muscle, as you can see by the plot here. So there's buildup of tension, and it is maximal, and it is sustained. There is no relaxation. So this sustained maximal contraction of the muscle is referred to as tetanus. Some of you may have come across patients that have tetanus, and you know this it's a result of I think they should be toxins from some kind of bacteria. I've forgotten, I cannot tell you now. And you know that this can, as a matter of fact, be fatal. If you've, especially in children, I remember when I was rotating through pediatrics, in the pediatric ICU at UTH, found the kid, who was having this tetanus. They had to sedate the kid because, from what I understand, it is actually quite painful, as a matter of fact. So this maximal sustained contraction of the muscle is going to, last as long as, you know, um, um, uh, as long as the action potential, until the action potential ceases to arrive or the muscle goes into fatigue. And I'm going to explain what muscle fatigue is a little bit later. So what you're looking at, ladies and gentlemen, here is basically the responses of the muscle in terms of tension to action potential, depending on the timing with which the action potential is arriving. So in the first, we've got the single twitch. So here we just have an action potential. It arrives, muscle builds up tension, but then relaxes and goes back to normal. You have another one arriving after the muscle has relaxed. So basically, it does the same thing. It relaxes. But you have the second one that is arriving before the muscle finishes relaxation. This one is called twitch summation, like we have said. Or you can have, an action potential that is arriving at the muscle, very rapid without giving the muscle any chance to relax. The muscle then will build up a lot of tension, which is said maximal tension, and which is sustained without any relaxation at all until it becomes fatigued or until the action potential ceases. And in this image, I'm basically showing you a patient who was drawn in medieval times, who was said to have this tetanus. Okay, so that you don't lose track of what we are talking about, basically we were talking about factors that will influence muscle tension. So from this we can see, when we talk about this single twitch, twitch summation, is basically the timing at which your action potentials are arriving with reference, to the contraction and relaxation of the muscle. So if it arrives too quickly and does not allow the muscle to rest, you can either have a twitch summation, and if it is, it's arriving in very quick succession without giving the muscle any chance to rest, you're going to have what is known as tetanus. And you can see how high this blue plot is going with reference to the timing, of the action potential that is arriving at the muscle. So, in short, the muscle tension in twitch summation is more than that of single twitch. The muscle tension in either single twitch and twitch summation is more in tetanus than any of these two, basically. All right. So we also have a phenomena known as the Trepe effect. Mr. Sizwe, sir, is the other gentleman with us?
Yes, no.
Okay, the other gentleman, is he with us?
No, it's not.
Oh, so we only have five participants.
Yes, we
Yes.
are just five today.
Okay. So who hasn't answered any question today? Okay, class rep, you can volunteer anyone. So I'm going to give you this question, and if you can't volunteer, you answer it yourself. So, class rep, I hope you like football. Have you ever wondered why those football players? I'm pretty sure you are watching this. Oh, but you said you don't watch football. Yeah, yeah, yeah. Anyway, but it's okay. But you're a health professional, so you should understand. Why is it then that those football players, before they are substituted, if the coach sees that they want to substitute number 10, because maybe he's getting tired or whatever reason, he has lost focus, before a player is pushed into the pitch, they're going to ask the player to do a few runs on the side. Do you know why that is so? You can volunteer anyone.
I volunteered Dolly to answer.
Yay!
I volunteered Dolly to answer that question.
Yay! Yes, Madam Jolly, can you tell us why? Madam Dolly doesn't want to tell us. So anyway,
Maybe Victoria can help us.
Madam Victoria, can you help us? And we're almost done, in case you're tired.
The reason why footballers run around before they are taken in the, maybe it's for their muscles,
Aha!
just
To
to
do what?
increase the.
Okay, so the, and it's just not at substitution. It's even before any athlete, um, 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 joins or goes to a competition. You'll have to prepare your muscles. You have to do a warm up. That's what we call it. So why is that important? It's important because of what is known as the trepe. Now this must be French. I am not French. So forgive me if I'm killing it. Probably borrowed from French. So this is a phenomena which has been noted to say, you know, the contraction, the subsequent contraction, so of a muscle, are going to be more or better than the previous ones. So meaning that if you warm up, the contraction of your muscles are going to be better than if you did not. So the warm up sort of acts as a preparatory measure, you know. And you can see in my plot here. So what do I have? So you can see that the blue plot represents the tension in the muscle. You can see that the initial stimulus. Let us assume that this is an action potential that is arriving at a muscle. The first action potential, you can see that it doesn't give us a lot of tension. The second action potential, the tension that it gives us is more than the first one. And you can see that even the subsequent one, the contraction becomes better and better as we go. So you can understand that if your footballer joined the pitch without warming up, the contraction of his muscles would be at this level when he could have gotten this level had he warmed up. So this is basically what is known as the Trepe effect. And the explanation for the Trepe effect is that when you warm up, what's going to happen is that there's the initial exercise is going to increase the release of your stored calcium in your sarcoplasmic reticulum. Additionally, it's going to lead to reduced slack of your elastic series components in your muscle. But also, when you warm up, you're going to increase your body temperature, which is going to help, that our enzymes, you remember that basic physiology we learned in, I think, secondary school, I think grade 9, that when you increase temperature, you know, the the rate at which your enzymes will work better at a higher temperature. But of course, there's a limit to it, after which, your enzymes will become denatured. But of course, we know that the body has mechanisms that it puts into place for that temperature not to be exceeded. So that's basically what is known as Treppe effect. So in Treppe effect, we are saying that the initial stimulus helps us to have better contraction, better muscle tension in subsequent contractions, basically. Okay, moving on. Among the factors that influence muscle tension is muscle optimum length. So, how to understand that is that we did say that a muscle fiber is going to be made up of actin and myosin in a sarcomere, in an arrangement, where they overlap each other. From this graph, you can see that this is what you expect to be optimal arrangement of your actin and your myosin. If they are too close to each other, then once activated, because they're already close to each other, there isn't room for movement and for the contraction to happen optimally. So the muscle tension, as you can see from the plot, will be suboptimal. However, in this optimal arrangement, you can see that we get this 100% build up of tension in the muscle. When the muscle has been stretched too much, you know, I'll give you an example. When somebody becomes incontinent, and then let us talk about, as you age, unfortunately for us men, you get BPH, that urinary obstruction. As a result of that, the bladder then is going to be, you know, basically becomes dilated. What will happen with the dilatation is that because the length of the muscles become too much, the if you look at this arrangement here, you can see that the actin and the myosin are barely touching. And if the actin and myosin are barely touching, meaning there are very few heads available to attach to your actin to bring the sarcomere, or rather to shorten the sarcomere. So basically making the contraction of this particular muscle fiber poor. So you can see that when you have stretched the muscle too much, the tension that you're going to build in that particular muscle is going to be suboptimal. And it becomes worse the more you stretch. If you look at B and C, you can see that C, because really the actin are very far from the myosin, the tension that you're going to build is very little compared to that in B, which still has has a slight significant portion of the actin and the myosin um, um, you know touching each other. Okay, moving on. So this is basically a summary of what we've already talked about. You can read it on your own. Okay, so like we've already said, we did say that the muscles, to be able to carry out this contraction or shortening of the sarcomere, they require ATP. We talked about the ATP is required in the power stroke. We also said that the ATP is required in the resetting process where the myosin and the actin are going to unbind. We also did say that the ATP is required to drive the calcium back into their storage place, which is the sarcoplasmic reticulum. Additionally, we did say that once the acetylcholine arrives at the membrane of the muscle fiber, what you're going to have is exit of the potassium from the muscle cell and entry of the sodium into a muscle cell. This requires energy, and this energy will come in form of ATP. But the question is, where does the muscle get this ATP from? So there are basically three pathways the muscle is going to get this ATP from. So the first one is basically, it is known as oxidative phosphorylation. You may know about glycolysis. So, but we know that glycolysis is inefficient. Um, when a glucose molecule enters glycolysis, you only produce about two ATP molecules. So it is quite inefficient. And we know that it leads to, from glycolysis, if oxygen is available, you know, your molecule is going to go into oxidative phosphorylation, which is the efficient one that produces more ATP than just glycolysis. However, if you do not have adequate glucose, what you're going to have is um, that you are going to have your pyruvic acid, which is a product of the glycolysis, which is going to be converted to lactic acid. Now, this lactic acid, it is an acid. It is going to alter the pH of the muscle and is going to affect, the working conditions for your enzymes in the muscle, and this will contribute to your muscle fatigue, basically. So apart from oxidative phosphorylation and glycolysis, the the muscle also gets ATP from a molecule called creatine phosphate. So this creatine phosphate, which is stored in a muscle, converts to creatinine, and when it converts to creatinine, it gives energy to a molecule called ADP. I think this adenosine diphosphate, and it gives a phosphate. Apart from energy, it gives a phosphate as well. So this molecule will be converted from ADP to ATP, and we know that ATP is the currency of energy for the body, and the muscle will be able to use it. So the interesting thing is that while oxidative phosphorylation is slow, it generates more ATP. This creatine phosphate and glycolysis, they are inefficient. They produce fewer molecules of ATP. However, they are quick to produce this ATP. Okay, so we can talk about the concept of muscle fatigue. So basically, muscle fatigue is when you have decline in muscle tension as a result of previous activity. So when there's muscle fatigue, your muscle will be unable to contract despite stimulation. So muscle fatigue may happen in a situation when you do not have ATP anymore. Okay, so shall we say it doesn't have to be a complete absence of ATP? So shall we just say, when there's depletion of ATP, your muscle is not going to contract anymore. We've already said in a setting where you have anaerobic respiration. So in anaerobic respiration, you are going to have a situation where there's inadequate oxygen. The inadequate oxygen is going to favor production of lactic acid. The lactic acid accumulation is going to result in a drop in pH, and we know that our body enzymes, including that of the muscle fibers, will not work optimally when the pH drops. So that will also impede the contraction of your muscle when there's production of lactic acid. It may also happen when you lose potassium via the sodium potassium pump and the body is unable to restore the balance. It may also happen when you have abnormalities at the neuromuscular junction, and there are several diseases that can cause that. Examples of those diseases include myasthenia gravis, a Lambert-Eaton syndrome, or botulism. Ladies and gentlemen, I also like to tell you about the concept of oxygen debt. So the concept of oxygen debt, guys, is basically the extra oxygen your body needs to take in to restore muscle chemistry back to resting state after muscle fatigue. Okay. With this extra oxygen your body needs to restore, your muscle chemistry to resting state, you know, the intake of this extra oxygen will result, in the conversion of the lactic acid, in the blood to pyruvic acid, which can be converted to glucose or may enter the aerobic respiration or the oxidative phosphorylation that we spoke about. It will also help to restore or replenish your glycogen stores. It will also help um, phosphorylation of your creatinine to form the creatinine phosphate, which we said are the molecules of energy that your muscle is going to use if it needed energy very quickly. And this extra oxygen is also going to rebind to your myoglobin. Very quickly, guys, I would like to tell you about the effects of exercise on muscle fibers. So, aerobic exercises, as most of you do, will result in more efficient muscle metabolism and resistance to fatigue. It's going to result in increase in capillaries. It's going to result into increase in mitochondria and myoglobin. You're also going to increase in the efficiency of your heart, lungs, and body metabolism, and that of your neuromuscular coordination. However, exercises such as weightlifting, which also known as isometric contraction, this will result in production of more myofilaments and myofibrils that is going to cause your basically your muscles to hypertrophy. You're going to increase your glycogen stores, and it's going to result in increase in your muscle size and strength. Basically, guys, that's what I prepared for you. Do you have any questions?
No question from me.
So it's either it was clear or this.
No questions for me.
I'm sorry.
And no questions for me too. It was clear.
I don't believe you.
I have a
Oh, it's
question.
true
Yes,
that.
yes, yes, yes. Question, please.
I would like to know what happens to those people who pass out when they're doing exercises.
Okay. I think that there are many reasons. There are many reasons why somebody may pass out. If, let us say, it could be a cardiovascular issue. So, what do I mean by that? It could be that the blood supply that is needed to you know to supply blood to the muscles as that person runs is inadequate. It could be, or it could be that maybe the autonomic nervous system is not working right, such that you have dilatation, an exaggerated dilatation of the blood vessels. And you know that if you have exaggerated dilatation of your blood vessels, you go into some kind of relative shock. You know how that happens. It means that your vascular compartment becomes bigger than what it should be. So your perfusion basically then drops to the brain, and then you end up with fainting. I'm not really sure if, let us say, as one exercises and they produce lactic acid, it is not usually systemic that it should go to the brain, that somebody should pass out. It's usually local at the muscle itself. So I would like to think that that is more of a cardiovascular system than anything else. So I don't think that there's one single explanation for it. In fact, because you could even, let us say, if we looked at the heart, even if you say that the sometimes you can have, you know, people like an individual with a conduction problem. So we are going to look at conduction issues, in the next semester, and such that when you stress the heart, it doesn't
Okay.
work right. So it could result in that. So I do not have, you can see that I'm everywhere. I do not have one specific answer. I think that it's a combination of factors depending on the individual. And I think really it has to do more with the cardiovascular system than the chemicals that are produced as the muscles are contracting, because it's not systemic but rather local.
Okay. Thank
Okay, so when are we having the test? When are we having the test? Tomorrow evening?
Can I propose?
Yes, please. Propose, please.
Sunday evening.
Sunday evening. Okay, that's fine. Sunday evening it is. The test is ready already. So even tomorrow we can do it.
you.
Sunday
Cecilia.
evening sounds okay.
Sunday evening is good.
And the good thing is that it's MCQ, so it's the same.
Yeah, I think Sunday evening, so that when you give us these notes, we can pass through them, because at some point some network was dropping. We're not getting everything.
Okay, now that's not a problem. And I have worked out some solutions, because I normally just give you these MCQs. It is difficult for me to give you those, scenarios. So basically, I do not expose you to what you're going to see in the exam. So I'm going to give you some sample questions from real exams at CRESO and solutions. So then you can, go through it in your own time, and then you can ask questions if you have.
That would be much appreciated, sir.
Okay. Thank you
100
so
kwacha
much,
per
sir.
copy. It's not free now.
No problem.
No
Even
problem,
200
sir.
if you want.
Even five.
1.5. That sounds good. All right. Not a problem. We'll talk
Yeah,
about it.
even 2000.
Even 2 pin. Hey, that sounds good now. No, it's fine. We shall talk about it when we meet. Thank you very much.
Okay.
Thank you.
Okay.
Tomorrow's lecture is supposed to be in